Hi, my name is Sumit and I am an insect biologist. I did my PhD in animal behavioral biology, and where I basically mainly investigated the proteins and genes involved in the mating behavior of fruit flies, very tiny fruit flies that you see hovering around common fruits like banana, oranges, etc. Now in today's topic, I'm going to answer two important questions. First is, why should we study insects? Why bother about them at all? And second is, how should we study them? What are the tools and techniques that we use in the lab to address important biological questions? I'm pretty sure that if you stick to your seats for the next 6 minutes, you'd be able to glean some information about how research is done in the lab. Alright, so coming back to the first question, why should we study insects? Well, if you look at the history, bubonic plague caused by fleas on rats, malaria caused by mosquitoes, West Nile virus, Zika virus. There are insects that cause damage and harm to human populations. But at the same time, there are insects like butterflies and other good insects that basically help sustain the agriculture by pollinating the flowers. So, if we understand the physiology and behavior of insects, we might be better adapted to counteract their populations in the future, may it be on any side, either increasing the population of bees or decreasing the population of mosquitoes. So it has a big impact on environment too, and this is why we should study insects. Now, how do we study insects? Thankfully, we have a representative insect called Drosophila melanogaster, a scientific name of a small fruit fly. And we have tons of genetic tools. Now by genetic tools, I mean we have the power to alter their DNA and study the specific functions associated with genes. Now if you have to think about this like assembling an IKEA furniture. It's like you read a manual and you, the reader, then assembles the furniture. In case of animals, the manual is the DNA, the reader is the RNA, and the furniture is the protein. DNA is basically present in all cells of your body. So if you take away a gene is a small segment of DNA, and if you don't see a behavior, we know that that particular protein or that particular gene is involved in that function. If you keep the gene back in that particular organism and the behavior comes back, we know, or we have documented successfully, that that gene is indeed the reason why that particular behavior was affected. So in short, we establish what happens or what the role of that particular gene in a particular behavior or physiology. That's the framework of a scientific question and scientific process. So using these tools, I investigated the role of a protein called takeout that is responsible for the mating behavior of a fruit fly. Now in doing so, I used a fly that didn't have that protein, so it was defective in behavior. I brought the good copy of the gene back in the fly, and I saw a rescue of behavior, and hence, like I said before, I established the role of that particular protein in the behavior. So now that we know that there is a protein responsible for the mating behavior, we can develop strategies, chemicals that can bind to this protein, and in a similar way can prevent the population of other insects. We can make these chemicals more specific to maybe each variety of insects. and hence target only them and not other insects which could be beneficial. So that was a very broad introduction to what I have done in the lab. I do feel that you might have questions, and if you do, please don't hesitate to reach back. In the end, I would like to give the credits to my advisor who guided me throughout this research project, and all the other people who were deeply involved in creating those valuable tools for us. With this, I would like to say goodbye. Thank you so much. Have a nice day.